Hello students, welcome back. See, today in this video we are going to study the structure of protein in detail. Okay, now see, protein structure is highly complex and they consist of one or more polypeptide chains arranged in them, okay, in certain sequence and all. Okay, now the structure of protein can be studied in 4 different levels. Fine. Primary structure, secondary structure, tertiary structure and quaternary structure. Okay, now as you move from primary to quaternary, the complexity will increase. So let us start with primary structure. Now primary structure of protein is the basic structure of protein, which consists of some specific sequence of amino acid linked to it. Okay, specific sequence of amino acids which determines the primary structure of protein. Okay, like for example, these are the amino acids and these amino acids, they are linked in a specific sequence. You know, one change in the amino acid can change its function because this sequence of amino acid is responsible for their biological functions. Okay, and therefore this sequence is extremely important. It is responsible for their specific functions. As we know, different proteins have different functions in the body and so on. Okay, so therefore primary structure, it represents the specific sequence of amino acids which further determines the function of the protein. Okay, let us see this with an example.
See, that's what I was talking about. The primary structure of protein, it represents the specific sequence. See, this is an example that normal hemoglobin consists of these amino acids in the proper sequence, okay, in this specific sequence. Now even if one of the amino acid is changed due to any means, like here this glutamine is replaced in normal hemoglobin by valine in valine, okay, then it will cause a diseased condition called sickle cell hemoglobin. Okay, and you have heard about sickle cell anemia. Okay, that will be the disease. So primary structure represents the specific sequence and the biological activity is decided by the primary sequence of the or primary structure of protein, which is very special. The secondary structure of proteins. Secondary structure, it gives the shape or conformation of the protein molecule, that is, the manner in which these polypeptide chains are folded or arranged. It was observed that the two common types of secondary structures are alpha helix structure and beta pleated structure. This diagram which you are seeing is of alpha helix structure. It is the most common form in which polypeptide chain occur. You can see that the chain is twisted into a right-handed helix with NH group of each amino acid residue hydrogen bonded to the CO group of an adjacent turn. Okay, and is the beta pleated structure. Beta pleated structure, here you can see all polypeptide chains, they are stretched out to nearly maximum extension and then they are laid side by side in a zigzag manner, as you can see in the diagram, and they form a flat sheet. When they are laid like this, they form a flat sheet. Now each chain is held together is held to two neighboring chains by hydrogen bonds, and these sheets, like one sheet is formed, then another sheet is formed in such a manner. These sheets are stacked upon one another to form a three-dimensional structure called beta pleated structure. Now as you can see in the diagram, two types of pleated sheets are possible. That is, in the A diagram you can see parallel, that is N-terminal and N-terminal are on the same side. So parallel polypeptide chains are running. And in the B diagram you can see anti-parallel, that is N-terminal is joining with the C-terminal. Next is the tertiary structure of protein. You can see over here in the diagram, the tertiary structure is showing an overall folding. So tertiary structure arises due to folding, coiling or bending of polypeptide chain, and hence it produces a three-dimensional structure. Therefore, it is responsible for the overall shape of the protein, or we can say in simple words that here further folding of the secondary structure occur. See, it is shown over here. Primary is the simple structure, secondary sort of alpha helix, and then the tertiary structure. In tertiary structure, the secondary structure is further folded. Okay. And the fourth one is the quaternary structure of protein. See, now there are several proteins which exist as single polypeptide chain, but there are some proteins which exist as a combination of two or more polypeptide chains, and these two or more polypeptide chains are called as subunits or protomers. And you know, these subunits can be identical or they can be different as well. And these subunits are held together by non-covalent forces such as hydrogen bonds, electrostatic interactions, van der Waals interactions, etc. So therefore, these quaternary structure, they refer to the determination of number of subunits. That is, they help us to determine the number of subunits and their arrangement in an aggregate protein molecule. Okay. Now you can understand all these structures with this diagram as well. Fine. Here you can see the primary structure, simple combination of amino acid. The colorful spheres, they represent the different amino acids. Then secondary structure, okay, then they are forming alpha helix and sort of things. Then tertiary structure, further folding is there. And in quaternary structure, you can see different colors, that is different type of subunits are being linked together.
So that was all about the different levels of structure of proteins, okay, primary, secondary, tertiary and quaternary, where secondary we have seen is being classified depending upon the further in depth about into this alpha helix and beta pleated, okay. So by the figures and diagrams, I hope you are very clear with these structures.